Hello everyone, welcome to the class of biotechnology. My name is Dr. Farah Maarfi. In today's lecture, we are going to discuss about the principle of chromatography, that is the partition coefficient, or you can say the distribution coefficient. But before we move forward, let's understand the basics of chromatography. That what is the word chromatography means, from where it is derived, and what is the history of chromatography. Actually, chromatography is a separation technique. By using the technique chromatography, you can separate out the component from any mixture. Suppose you have a sample. In sample, you have different components. Suppose you have component A, B, C, D, E, right? And you want to separate the component D out of this sample mixture. So by the means of chromatography technique, you can separate out this component D, and by achieving the component D, you can go for the characterization or whatever study you want to perform. Chromatography word is actually derived from the Greek word that is chroma. Chroma means color and graphy represents the writing. Graphy means writing. So whole together, chromatography means color writing. So by means of the color writing, you can easily separate out the components out of mixture, right? Whole together, chromatography is a separation technique, or you can say it is also an identification technique by which you can characterize it, or you can study the component present in any mixture, or you can separate out the particles through it, and you can go for the characterization. Basis of all form of chromatography is distribution coefficient, or you can say partition coefficient. Actually, distribution means how any particle, any mixture or any component is distributed throughout the phase. That is, what is the pattern of distribution, how they are being distributed throughout any phase. This technique is first used by a botanist named Michael Sweet. Actually, he wanted to separate out the component of the leaf. So for that, he has taken the leaf extract. Leaf extract. This leaf extract actually carries chlorophyll, xanthophyll, and what else? That is carotenoid. So Michael wanted to separate out these component of the leaf, right? Initially, it was present in a mixture that is the leaf extract, right? So from the leaf extract, he wanted to separate out each component. So to separate out these component out of the leaf extract mixture, Michael has taken a column with a stopper at the other end here at the bottom. He held this column with the help of a stand and put a little bit cotton at this part here and filled the column with calcium carbonate, followed by a buffer solvent, and this buffer solvent is organic in nature. He filled the column with a calcium carbonate, and after that he has taken the solvent so to equilibrate the column with it, right? The purpose of this solvent is to provide equilibration to the column. Initially, Michael has kept this knob in a closed position, and he has put it 3 to 4 beakers at this point at the bottom of this column, so to collect out the separated components in a serial manner. After providing equilibration to this whole setup, Michael now opened this column and he allowed the leaf extract to pass through this column. After few minutes, or you can say within a few minutes, he observed different colors of bands in the column, like this: green here at this position, then orange and yellow color at this area. So as soon as he encountered the separated different bands, he started collecting each of them here at the bottom in the container. Suppose this is container A, B and C. So he collected the green color component here in the beaker A, then orange in this beaker, beaker B, and then yellow in this beaker, beaker C, separately. Like he started collecting each of them separately. These days, all the chromatography technique is based upon this technique of separation and is widely used in advanced way. So this was the little bit about the history of the chromatography and how easily you can separate out a component from any mixture. But this is specific to a few type of components or few type of mixture. Suppose you have a sand in a water, then you did not require chromatography for that. You can separate out the sand particles only by the means of that filtration technique, right? Let's discuss about the basic principle of chromatography, that is distribution coefficient. So now question arises, what actually the distribution coefficient is, or you can say what actually the partition coefficient is, right? These both are the almost same term and is represented by KD. KD here represents the distribution coefficient. But before we move forward in terms of the distribution coefficient, we should know that all chromatography system consist of two phases, that is the stationary phase and a mobile phase. These two phases are immiscible. They are immiscible, that you cannot mix them together, right? You can use solid, gel, liquid, solid liquid mixture as a stationary phase, and for the mobile phase you can use a liquid or gases as a mobile phase. Mobile phase are mobile, that it can move from one position to the another position, right? Suppose this is a stationary phase. Stationary phase is actually is immobilized, that you cannot move the stationary phase. It is immobilized, it is fixed at a particular position, and a mobile phase is passed through it, right? Mobile phase can pass through the immobilized stationary phase. Distribution coefficient describes how a compound, or you can say analyte, is distributed between these two immiscible phases, that is stationary phase and mobile phase. That means how a particular analyte or a compound is distributed between these two immiscible phases. Specifically, it quantifies the affinity of the analyte or the compound for one phase over the another phase. Let's understand this by an example. Suppose this is an interface of oil and water. Oil and water. And you have analytes here, right? So it depends upon the distribution coefficient, that which phase this particular analyte prefer over the another phase. If it goes to this side, that means the analyte prefer the oil over water. This is a phase A and this one is phase B. So if it goes like analytes to this few and few of them goes to this side, so the one that goes into the oil phase describes the affinity of that particular analyte for the oil over the water, and similarly the analytes that goes towards the water describes the affinity of the analyte for water over the oil. Distribution coefficient actually quantifies the affinity of the analyte for one phase over the other phase. KD also influences how quickly an analyte moves through the chromatographic system. Suppose this is a column and this column is coated with stationary phase. Stationary phase, suppose here is a solid. Stationary phase can be solid, liquid, or gas. This time, suppose this is a solid, and you coated this column with a stationary phase, and you have an analyte A, B, C, D. This is a mixture of analyte. Mixture of analytes, right? When you mix this analyte with the mobile phase and passes this mobile phase through this column, so as soon as the mobile phase come out of this stationary phase, the analyte that have a more affinity towards the mobile phase will come out first. Suppose analyte B is having a more affinity for the mobile phase than the stationary phase. Then whenever the mobile phase comes out of the column, analyte B will come along with it. Analyte that comes out of the column is analyte B. Suppose after that the C, analyte C is having affinity for the mobile phase. After that, in the second round, the mobile phase carries the analyte C along with it. So the second analyte that will come out of the column is C. So first one is B, then C. And analyte A is not having affinity for the mobile phase, but it is having a affinity for the stationary phase. At this time, analyte A will stick with the stationary phase here in the column. So in this way, you can separate or distribute, or you can understand the distribution pattern of the analyte between the two phases. During chromatographic separation, the analytes continuously pass back and forth between these two phases, so that the difference in their distribution coefficient results in their separation. Means how a particular analyte is being distributed between the two phases is the principle behind the separation of the particular analyte. The distribution coefficient for any analyte can be calculated by using this formula, that is the concentration of the analyte in phase A divided by the concentration of the analyte in phase B. Kd here represents the distribution coefficient. And a statement that we should consider in reference to the chromatography is like dissolves like dissolves like. This is very important statement. You should keep this statement in mind so to understand the whole concept of each type of chromatography in much better way. This statement actually depicts that the substances with a similar polarity tends to dissolve in each other. That is, the substance with similar polarity tend to dissolve in each other. Dissolve in each other. That means if the compound or a substance is polar in nature, then it will only get dissolved into the polar solution, right? Or if the substance or the compound is non-polar in nature, then it will only dissolve in non-polar substance solution. Example for the polar substances or the polar compound that can dissolve in a polar solution of the solvent is the ethanol and water. You can readily dissolve ethanol into the water. Another example is salt. You can dissolve salt in water very easily. Another example is sucrose in water. Sucrose in water. Ethanol, salt, sucrose and water all are polar in nature. They all are polar in nature. When we talk about the non-polar solution, then the examples are hexane in benzene. You can dissolve hexane in benzene. Another example is you can dissolve iodine into hexane very easily. Next example is oil in gasoline. Oil in gasoline, or the sulfur. You can readily dissolve sulfur into the carbon tetrachloride. Carbon tetrachloride. Hexane, benzene, iodine, sulfur, carbon tetrachloride, oils, gasoline, all are non-polar compounds or non-polar substances. So the polar compound only dissolves into the polar solvent and the non-polar compound only dissolves into the non-polar solvent. But the polar and the non-polar compounds do not dissolve in each other. This is the basic and the most important concept of the chromatography. If you understand this concept, you can easily figure out that how to separate a particular compound by using a particular chromatography. So that was all about the chromatography, what actually the chromatography is, and what is the basic principle behind it, what is the history of the chromatography. In the next lecture, we will study the different type of chromatography. Till then, thank you so much. And if you like this video, please like and subscribe. And don't miss to hit the notification bell so that you cannot miss any of the future video. Thank you so much.